So I'm Professor Sarah Howie, and I work in this building in the Centre for Inflammation Research. And there are 3 University of Edinburgh buildings on this campus. This one, the Queen's Medical Research Institute, the Chancellor's building next door, and the Scottish Centre for Regenerative Medicine, which is on the other side of the hospital. And within this building, QMRI, there are 3 research centres: the Centre for Reproductive Health on the ground floor, Centre for Inflammation Research on the middle floor, and the Centre for Cardiovascular Science on the 3rd floor. And both speakers tonight are actually from the Centre for Inflammation Research within this building. And we're very pleased tonight to have Professor Tim Walsh, who's a clinician and Professor of Critical Care, and Professor Adriano Rossi, who's a scientist and Professor of Respiratory and Inflammation Pharmacology. And I just have to tell you that this series is sponsored by the Medical Research Council.
So thank you all for coming. This is the title we've given ourselves, and perhaps at face value you don't really understand what we mean. So what I'm going to try and do for the first 20 minutes or so is tell you about the type of patients that we need to develop new treatments for, and try and convince you and explain to you why this is really important to these patients and also to the health service, because as I'll show you, these patients cost a lot of money as well as being very sick because their recovery is very slow. I work in an intensive care unit, and that picture there is a picture of the intensive care unit in the Royal Infirmary next door, and that's where I practice my clinical work. And I'm a researcher as well. I mainly do research, clinical research with patients, trying to do clinical trials and understand diseases in the NHS, in the clinical environment, and work about recovery. So I'm going to start off by telling you about a real patient. And I looked after Louise, who at that time was 25, in October 2009. And she was admitted to this hospital as someone who previously was essentially entirely well. She had very mild asthma. She just took the occasional puffer, but she was a completely well, fit person. But she came into the hospital with a fever, she had a temperature, and she was feeling breathless. And when she came in, because she was breathless, and the concern was there was something wrong with her lungs, she had a chest X-ray done. If you haven't seen a chest X-ray before, over here with my pointer, that's the right lung, and that looks fairly normal because you can see blackness throughout where the lungs are, because the lungs are full of air, because we breathe in and out through the lungs. So on an X-ray, they look black, the way we take X-rays usually. But I expect you can see on this side, there's something going on here. This shadow in the middle is the heart. And there's some white shadowing in the lower part of her left lung. And in a young person with a temperature who's feeling breathless and feverish, that's almost certainly a chest infection or a pneumonia. It means the same thing. So she was started on antibiotics to treat that infection because it was quite possibly due to a bacteria, and she was given some oxygen and she was admitted to the ward. But unfortunately, over the next 2 or 3 days, things got worse. And this is Louise's X-ray 3 days later. And I'm sure you can see that now this whiteness has spread to both sides of the lungs, and it's increasingly affecting all of the lungs, with just some parts up here that on this type of X-ray there's blackness. In other words, you can still detect the air spaces in those lungs. So what she now had was what we would call a condition called ARDS, or Acute Respiratory Distress Syndrome. So it's quite a descriptive term because the people who have it are struggling to breathe, and they can't get enough oxygen into their blood across their lungs through their own breathing. So there's a definition of what ARDS is. It's something that comes on rapidly. It's not a chronic disease that people develop over months or years. It's something that happens in a few days. It affects both lungs. So in this condition, Louise had a chest infection and a pneumonia that developed and affected both of her lungs, from having initially only affected one. And it results in failure of our respiratory system, our lungs, and our ability to breathe and get oxygen into our blood and carbon dioxide out of our body into the air, which is the primary function of our lungs. So it causes what in medical terminology is called hypoxia, which means a lack of oxygen in the blood and the body. Now, ARDS is not a disease like pneumonia is or cancer is. It's a syndrome, which means it's a complication of lots of other diseases. So you don't get ARDS on its own. There's always something else that causes it. So before we move on, I just want to show you, and if you know all this, remind you what normal lungs are like. So here's a normal picture of a left lung. And here's the trachea, or the windpipe, which we breathe in and out of, going into a main bronchus, a division, and then into lots of smaller airways that transmit air in and out as we breathe. The important thing happens in all this lung tissue. And if you magnify a tiny bit of this lung enormously, you'll see what is sometimes termed sort of bunches of grapes. And these are called alveoli. These are tiny microscopic air spaces at which we actually exchange gas, oxygen and carbon dioxide, from the lung into the blood. So here's looking down a microscope at a piece of normal lung tissue, all these air spaces called alveoli, and each of these are supplied by a tiny airway that we're breathing in and out of. If you go even closer in, you can see that between these alveoli you have a blood vessel, a capillary, and along this capillary all the cells that are in your normal blood move. And those include the red blood cells, and they're the ones that are exchanging oxygen and carbon dioxide, but it also includes white blood cells, which we have in our circulation, which are involved in immunity and fighting infection. Now, I just want to ask you a question. How big do you think all my lungs would be if all those alveoli could be laid out flat in this room, just my lungs? Just put your hands up. How many people think it would just be this front row? If we made the surface area of just this front row, how many people think it would be that? It's only me. Okay, one person. Everyone's got to express an opinion. How many people think it would be the first 4 rows? So going back 4 rows, along there and down there. How many people think it would be that? Okay, a few more people. And how many people think it might be closer to everything in the middle here, going right to the back? Ah, you're all very good. So as you can see there, the actual surface area of an adult human lung is enormous if you look at that area that's available to exchange oxygen and carbon dioxide. It's about 8 by 8 meters. So what happens in this condition called ARDS? So it's an inflammatory condition, and we're going to hear more about inflammation, particularly from Adriano after myself. So this is a picture of one of these alveoli blown up to enormous magnification and in a diagram format. So here's the airspace here, and the air would come in and out of here into all these millions and millions of alveoli. And in this area, there's some important cells. We have what's called a type 2 alveolar cell, and that cell is really important because one of the important things it does is it produces a detergent called surfactant. Not a detergent that you get for your kitchen, but a physiological detergent that our body produces. And that's really important because these tiny little air spaces would collapse because of the surface tension within them if we didn't have something in our bodies to reduce that tension. And so these cells produce this surfactant all the time. We have these lining cells across which the oxygen and carbon dioxide move, and we have the blood cells here receiving it or bringing the carbon dioxide back. And then here within the airspace we have something called an alveolar macrophage, and this is an inflammatory cell, and it sits there, and one of the things it does is it's looking out for infection, and it's trying to keep the lung free of infection, and it's fighting bacteria amongst other things, and it can produce substances that can kill bacteria. Now what happens in ARDS is a bit of chaos. So we see lots of things which we can't go through in detail, but the key events we see this alveolar macrophage switched on and starting to release substances to try and kill bacteria, and also those substances start to bring help and start to bring other inflammatory cells, in particular neutrophils, which we're going to hear a little bit more about which start to move into the lung spaces to try and apparently fight what's going on, because the cells think there's something going on, they should be there. The other thing that happens is that this membrane becomes thickened, and in particular it becomes leaky, so that fluid starts to go into these air spaces as one of the reactions. And in addition, this alveolar type 2 cell stops producing surfactant very efficiently. And these lining cells get damaged and may die, and you get a sort of a membrane here, which is sometimes called a hyaline membrane. And all those processes are explaining why their chest X-ray's gone white, because things are filling up with fluid and inflammation. So we can visualize that in real patients. This is a normal chest X-ray, and this is a CT scan. So imagine you're sitting at the feet of this patient, and you're looking up from their feet towards their chest. This is a slice created by X-rays that reconstructs what this slice across someone's chest would look at just above the heart. And this is a normal CT scan, and I've already shown you this is the alveolus at that microscopic level. Now Louise, at the stage we've got to, has now got some changes. This isn't her X-ray, but this is an early ARDS X-ray. And now the CT scan looks very different. You can see some areas don't have any air in at all. They're full of fluid and they've collapsed. And some areas have got areas of collapse and fluid and inflammation going on. And down here in the individual alveolus, we're seeing fluid filling up and inflammation starting in these air spaces. And then in the most severe forms, the chest X-ray almost becomes completely white. And the CT scan has major areas of whiteness and very small areas of darkness in which oxygen can get into the blood and so on. And the alveolus is severely damaged, and there's very small surface area, if any, in some of the alveoli for oxygen and carbon dioxide transport. So I want to illustrate what it must feel like, or what's happening in the lungs. You may wonder why this has been sitting at the front. Because if you imagine that sponge, which is full of lots of little air spaces, imagine that is your lungs. And that fluid's now going to start filling it up. So let's put it in there and imagine that inflammation is going on. And the other thing I want to do is give some of you some balloons. And anybody who can blow up one of these balloons, I'll give a £5 to. If you can blow it up, I'll give you a £5. I'd have one up here, or two even. There we go. So try and blow up that balloon, because imagine, although it's a long thing, it's not a round balloon, imagine that is an alveolus that has lost its surfactant, and therefore it's tending to collapse down because of all that surface tension. Now, nobody give themselves an injury trying to blow it up, please, because you won't be able to. So if you want to blow up a balloon in that situation, you've got to have some sort of pump. But even when you pump it up, it's going to carry on trying to go down, as that one does. The other thing is, our lungs have now filled up with fluid. So imagine what it must be like trying to breathe through those. So that is what's happening in a patient with severe ARDS. And that's what's happening in our patient Louise, who we looked after in 2009. Nobody managed it, did they? So what are some of the causes of this condition, this inflammatory condition in the lung? So probably the most common is the one I've got on the top left, where it starts in the lung and it's due to some sort of infection, like a pneumonia, which is usually due to bacteria, that then something triggers off that the whole lung gets affected by an overwhelming inflammation happening over and above the infection. In other words, it's an abnormal inflammation. We don't want it to happen anymore. It's damaging the body, not helping it. If you actually aspirated your stomach contents, that can cause it. Viral lung infections can also cause it in rarer situations. If you rescue someone from drowning and they've swallowed or inhaled rather water, that can cause it. And if you are in a fire and inhale smoke, that can cause it. But interestingly, we see lots of patients who develop this condition when the primary problem wasn't in the lung at all. And any severe infection in other parts of the body can cause this inflammatory condition in the lungs, even though the lungs didn't have the infection. And similarly, if you're in a major road traffic accident and have lots and lots of injuries and have to have lots and lots of blood transfusions, you can get it. And then there's some other conditions which we commonly see that cause inflammation in the body, but affect the lungs as one of the consequences. So here we've got Louise. Now there's a real picture of Louise because Louise is happy for us to use these pictures because she helps us try to explain to people what her experience was like and why it's important to do research. So don't be worried I'm showing something about a patient that she shouldn't, because Louise would be very happy for us to do that and has given us permission to do that. In fact, she would be here, but she's in the Dominican Republic on holiday, which is very nice. Now Louise actually, although she had a bacterial chest infection, what we realised was, and some of you, many of you will be old enough, some of you may not be, to remember that in 2009, everything in the news was about swine flu. And we saw a small number of people, about 1 in 10,000 people who were infected with swine flu, who developed this really, really severe reaction to it. So this is what's called a viral pneumonitis. It was very, very rare, but from other research we did here in Edinburgh about the same time in collaboration with other people, we know the virus was all the same, and it was about your own genetics and the way you reacted to that infection, which determined whether you got the ARDS. And that's very important with inflammation because we know that different people can react with inflammation in different ways. Now what we had to do with Louise was all sorts of things in the intensive care unit to keep her alive. Because as you can see up there, she had to have a tube put down and she was on a breathing machine to help these swollen, heavy, inflamed lungs keep her alive. Now, she was actually in the intensive care unit for many, many weeks. And during that time, here's my balloon, we were having to use lots of pressure to actually keep her alive on the ventilator. And that's actually quite tricky. And I can't do it now. My pump is broken. But one of the problems that she suffered... Oh, now I've gone deaf as well. Is that when the lungs are really heavy, imagine that sponge, we're having to apply quite high pressures with a machine to keep those lungs open. Imagine it with the sponge there and the balloon that some of you are trying to blow up. And one of the complications she developed was what's called a pneumothorax or a collapsed lung. And you see there over where the arrows are, you can see an area that doesn't have any shadows. Hopefully you can all see that. And that's that's a collapsed lung, which is one of the complications we see over here. That was about 2 weeks later. And the other thing she developed as a common complication was she developed another infection in the intensive care unit because she was really sick and because people with tubes in all sorts of places are at risk of developing further infections. And then we call that a ventilator-associated pneumonia, more shadowing there, another infection superimposed. And you can see here's her chest drain. That's the drain we put in because of that collapsed lung a little bit earlier. So here she is again. She needed to be in the intensive care unit on the ventilator for a month. Now, not many of our patients need that, but some people do. And you can get the impression, seeing how she was then, that she was exhausted, she was weak, and it took her a long time to be strong enough to come off the breathing machine. And as she gradually woke up, you can see how weak she was, because this was her attempt to write at that time. And she found it very difficult to do that. So how common is this condition? Well, there's various estimates, because it depends how you exactly look for it, but maybe about 13 to 23 people per 100,000 of the general population each year. You may not think, well, that's not all that many, but it has a huge effect on those people's lives. But of course, very importantly, depending on exactly how you estimate it, 25 to up to 40% of people die with this condition and the other associated conditions they have with it. And its effect on long-term health is very significant. And as I said at the beginning, in terms of the cost of this illness to that person and the health service, it's enormous, very high. But we have very limited tools at the moment to work out exactly what's going on in those lungs at different times. So it's very difficult for us to know exactly how we might modify the inflammation, because we might have unintended bad effects if we do the wrong thing at the wrong time. So what therapies do we have? This isn't an exhausted list, but because it's all about inflammation, we believe that therapies that we know change inflammation and generally tend to dampen it down, we believe they should work in this condition. Now, many of you have heard of steroids, and steroids have been tried in ARDS, and the problem is they have beneficial effects and harmful effects, and overall they don't help. Statins, which are very widely used drugs in cardiovascular disease, also have anti-inflammatory effects. They don't work. Neutrophil elastase inhibitors, that's a mouthful, but you'll hear more about these cells. Essentially, we've explored a number of different drugs which change inflammation or the way fluid accumulates in the lungs in different ways. The other thing we've tried is all sorts of different technologies to be as gentle on the lungs with the machines we use as possible. Because we know that if we damage the lungs with the machines, we just make inflammation and the damage get worse and worse and worse. So which of these have worked and which of these haven't? So generally speaking, we know that the way we manage the patient in the ICU with the machines and the very fine tuning of that is what's making a difference. But none of these drugs, and there are others, have made a difference yet in our trials, which is very frustrating. The only one that has been associated with benefit is paralyzing the patient's muscles completely, which you do with an anesthetic drug, for the first few days. But that's nothing to do with inflammation. That's more about these drugs probably reducing the damage from the ventilator by relaxing the patient. So we're frustrated by our lack of process. So I just want to show of hands. So in someone like Louise, say 3 to 6 months, let's say 6 months after surviving this terrible illness she had, put your hands up if you think this person would still suffer from muscle weakness and fatigue. Okay, lots of people think that. How many people think there's an increased risk of being anxious and depressed? Yeah. How about memory problems? Yeah, a few hands there. Joint stiffness. Yeah, a few hands there. You're getting the message. Post-traumatic stress disorder. Yes. So all these major effects on health are associated with a severe prolonged ICU admission, of which ARDS is one of the major causes. So just in my last couple of minutes, I want to tell you a little bit about a form of research called epidemiology and what it helps us with. So epidemiology is using large amounts of information about patients to study the factors that might contribute to a disease or factors that might influence the outcome from a disease in general terms. And this is work done by Naslone in our group. And in Scotland, we're very lucky because we can link all the health data sets in Scotland for the patients who've been in hospital. We're one of the few countries in the world that can do that, so we can find out what's happened to people. And all the patients who are admitted to intensive care in Scotland are all collected into a single database. And we link that to national databases that tell us about all their hospital admissions before and after, and whether they survive and how long they survive for, because that's all collected as part of standard data collection in Scotland. What we did in this study is we took patients who'd been in intensive care, not necessarily with ARDS, but just admitted to intensive care, and we matched them to some hospital patients who were very similar in many ways, but they weren't in intensive care, and the general population. And then we looked at the patients who actually survived to hospital discharge as well, because we know many people, as I told you, actually die during their ICU admission. So this is what's called a survival curve over 5 years for patients who, in this position, came into intensive care, and this is the proportion survived. And you see lots of patients who are going to die when they're critically ill die very quickly in the first few weeks, unfortunately. Then after that, some more of them die over time. And then this is the number of patients in hospital who are tried to be matched to this other group, and then this is the general population, I guess all of us sitting here, but matched for age and gender and a few other things actually as well. If you only take the patients who survived, knowing that lots of patients die from that acute illness, the important message of this graph is that you still see that what happens to these patients after they've survived the illness and gone home is still very different. In that about 20% more patients who've required to be in intensive care die over the next 5 years, even after you've tried to match them in lots of ways, and about 10% more than other general hospitalised patients. And if you look at the factors that are associated with a particularly high risk of that extra chance of dying, even if you get home, then one of the strongest predictors is that you had ARDS in the ICU. That's what that graph shows. So there is Louise's X-ray 5 months after she was admitted to intensive care in October 2009. It essentially is back to normal. So that terrible inflammation she had, having survived the illness, has gone away. And that was Louise in intensive care. And that was Louise about 2 months ago. That's me, in case you don't recognise me. About 2 months ago, when she came and helped us with another similar event to this and actually spoke about her experience. And you don't need me to tell you that there is someone who essentially returned to very good health and normal life. And she essentially has no long-term effects from this. So I'm going to finish there, and I'm going to hand over to Adriano to tell you more about the inflammatory side of things.
Good evening, ladies and gentlemen, boys and girls. Welcome. Thank you, Sarah, for the introduction. Thank you, Tim, for your presentation. What I'd like to do tonight is strip back some of this inflammation that you've heard about right back to fundamentals. We're interested in lung disease. I'm a scientist based in the MRC Centre for Inflammation Research, and we're interested in lung disease in general, and particularly ARDS, as Tim mentioned. I would like to put inflammation into perspective. And what is inflammation? Well, inflammation is really evolved to deal with invading organisms. These are bacteria, it could be viruses, it could be fungi, and there's millions upon trillions of these within the body. And how does the body deal with that infection or potential infection? Well, it has these blood cells, these white blood cells. Tim mentioned the neutrophil. That's the particular cell that I've been interested in. This is the neutrophil, and it's on top of some red blood cells. So this cartoon here highlights inflammation. Inflammation's been known for a long time. People have known some of the cardinal signs of inflammation. If you have a cut or heat or burning here, you will have heat. You will feel heat. There'll be redness. There'll be swelling, and there's also pain. And if the inflammation is so severe, you may have loss of function of a lung or perhaps a limb. So inflammation is very important. So what is inflammation? Well, it's a complex biological response to harmful stimuli. Stimuli such as bacterial infection or some form of trauma, a cut, a burn, etc. It's normally there to protect you. It's normally beneficial. So it basically gets rid of, for example, the infectious agent, and it's there to defend you, so it's involved in host defense. It's also involved in tissue repair. This is an area that people are more and more interested in. What does inflammation do to tissue repair? Well, it's very important. However, as you've heard from Tim, that uncontrolled inflammation can lead to inflammatory diseases. I've listed some inflammatory diseases of the lung here, but there are many, many more inflammatory diseases where inflammation plays an important role. In fact, it's actually difficult to think of any disease where inflammation doesn't play an important role. It involves many substances or mediators that are released within the body that mediates or controls the inflammatory response. And there are many cell types that also do the same thing. They control the inflammatory response. But it's especially the white blood cells that are key in regulating inflammation. People are beginning to realize that inflammation is really important, not just for the classical inflammatory disease that you can think of, ARDS, rheumatoid arthritis, asthma, etc., but also other diseases, cardiovascular diseases, heart attacks, for example, or neurological diseases such as Alzheimer's. Inflammation plays a key role. And what people are beginning to look at now is the resolution of inflammation. Inflammation normally resolves, it goes away, and there are a number of processes which are involved in that resolution process. And this is where we are interested in our lab, trying to understand the resolution of inflammation. If you can make unwanted inflammation go away using normal responses that are located within the inflammatory response, you'll be able to perhaps come up with new therapies for diseases. So stripping it right back to fundamentals here, this is a white blood cell, this is a neutrophil, and the neutrophil has evolved to deal with invading organisms. It can do it by a number of different processes. This is a process of phagocytosis where it engulfs or eats some of these invading organisms, some of these bacteria, for example. It can also undergo this process called degranulation, where it releases toxic products which can kill and damage these invading organisms. However, if that is uncontrolled, it can also damage the tissues of the host, your cells, for example. And also, this has recently been described as a kind of a strange way for a white blood cell to actually kill and trap an invading organism. This is called netosis. It's a process where the DNA, actually, it's the DNA that throws out a web of DNA plus other proteins which traps and engulfs the bacteria. Then other white blood cells can come along and gobble up all those harmful bacteria and get rid of them. So I just want to ask a couple of three questions to you. This is a very simple one. How many people are there in the world? Hands up who thinks there are 75 million. Not many. It's a very simple question. How many think there are 750 million? Not many. Nobody, in fact. How many think there's approximately 7.5 billion? Yes. So how many think there's actually 750 billion? Nobody. That's good. Not yet. So we're approaching 7.5 billion. This is just to put things into perspective. That's the answer. Now, this is another question. How many human cells are there in a human being, in an individual like myself? Are there A, 40 million? It's quite a big number. B, 40 billion? Nobody. What about C, 40 trillion? Okay. What about D, 400 trillion? Okay, it was split 50-50. The answer is actually 40 trillion. This is a big number. Look at this. Now, the next question is, how many microorganisms or bugs live in one human being? Okay. Oops. That was deliberate. Yes, the answer is 40 trillion, okay? So there's about at least 10 to 100 times more bugs in your body than you have cells. Just imagine that, okay? You're just a mass of bugs, essentially. And actually, people are beginning to realize that the ecosystem of your body and the bugs is very important. And it might actually have an impact on how you behave, and how you live, and how your whole metabolism is. So there's a big interest in the microbiome, which might actually be very important. We're actually being described as a superorganism, which is actually quite interesting. So here we go. This is this process that we've begun to look at. This is called netosis. So you have your white blood cells. It throws out this DNA and histones and various other proteins, traps these bugs, and then other cells can come along and engulf the bacteria. How do we do this? Well, first of all, I need to look for healthy volunteers like yourselves. Take blood. This is one of my former PhD students. She's very happy giving blood to me. Surprising. And what we do is we isolate those white blood cells. We purify them from the rest of the cells in the blood, the red cells and other types of cells. And then we do experiments on them. Okay? So what we've done is we actually isolated these cells and looked for these nets that are produced. And as you can see here, these nets are produced quite rapidly upon exposure, for example, to bugs. We throw out these nets and they can trap bugs, and then other white blood cells can come along, gobble them up and get rid of them. And we're trying to understand the process involved because we think they're important not only in defense of your body against infection, but also if this is uncontrolled, it might actually affect disease processes. So we're interested in that. We asked the question, how ancient is this phenomenon? It was only discovered about 10 years ago. So we actually, with Callum Robb in the lab, decided to go way back into the evolutionary scale, look at ancient organisms. So we went to the beach, we had a look at some of these shore crabs. We got some mussels. These are mussels. Some people eat them. But nobody really eats this. This is a blob. It's basically a jelly. It's a sea anemone. And the interesting fact about these organisms, they have white blood cells called hemocytes. And these hemocytes can actually defend the organism from infection. So what Callum and various other people did in the lab, they took the white blood cells from these organisms and looked to see if they produced nets. And lo and behold, they did. So we actually concluded that this process here of extracellular traps being produced was important in defense against those organisms. This is the highly magnified, several thousand times. You can see that these things are thrown out to trap bugs. So, back to the resolution of inflammation. So, the resolution of inflammation involves, first of all, you have your injury or your infection. Inflammatory response occurs. You have mediators, which I've mentioned. There's a whole host of mediators, perhaps over 100 mediators. So it is a complex process. Mediators are generated, and then inflammation occurs, and then with time, this time can vary, it will resolve, and it will return the structure of, for example, the lungs or the skin back to normal. And there are a number of processes involved here. And the process that we're interested in is a process called apoptosis. And apoptosis was named after leaves falling off a tree, but what it is suicide of the cell. So once these cells, especially neutrophils, have done the function, have done what they're supposed to do, remove invading organisms, they undergo this process of suicide, cell suicide called apoptosis. So what is apoptosis? Well, this is probably the very first picture ever taken of apoptotic neutrophils by Chris Hazlitt and John Saville. And what you can see, these are all one particular type of white blood cell. These are neutrophils, these are normal neutrophils, multi-lobe nucleus. And then when they become apoptotic, they become a little bit smaller. The nucleus is a bit more condensed, so they stain more darkly, and the membrane is still intact. So what we hypothesize, if you can make unwanted cells, once they've done the business of getting rid of infection, into apoptosis, you'll be able to resolve inflammation quicker. So this is what happens when inflammation doesn't resolve. You essentially get chronic inflammatory conditions or severe inflammation like Tim mentioned. You have inflammation starts, maybe you have too many of these mediators, too many inflammatory cells. Perhaps the cells don't undergo apoptosis properly, and they're not cleared by other white blood cells called macrophages. Tim mentioned macrophages. So what happens to these apoptotic cells? They're recognized by macrophages and removed in a silent, non-inflammatory manner. So this chap here is a neutrophil. I thought he was happy, but somebody pointed out yesterday that it's not a very happy chap. And the process of apoptosis. So this video here are neutrophils, human neutrophils taken from blood, the healthy volunteer, bathed in a solution of green dye and red dye. And what you'll see here, that this cell here will bleb quite nicely, very rapid. It's obviously speeded up a little bit. It will go green, these will all go green, and eventually they'll go red. This is the process of apoptosis, and because there's no macrophages to remove those cells, they will undergo this process called necrosis, where this red dye enters the cell. So this will go red, red, and red. And when they become apoptotic, they look like this. Essentially, the membrane is still intact. It has all these toxic contents still within the cell, and the nucleus is condensed. So what we hope to do is try and drive apoptosis to resolve inflammation. And how do we do this? Well, we kind of stumbled upon these, a family of enzymes, of proteins called cyclin-dependent kinases. There's at least 11, 12, 13 types of these proteins. And they're known to be involved in cell proliferation, and also some of them are involved in the production of mediators. So we almost accidentally tested some of these drugs in the lab, cyclin-dependent kinase inhibitors. Some of them are actually quite good at inhibiting cell proliferation. So in cancer, cells proliferate unwantedly, too rapidly, and the cancer takes over the body. So some of these drugs are in clinical trials for cancers. But what we found was that if you use some of these drugs to inhibit the production of some of the mediators, it affects specific white blood cell apoptosis, cell death. And then we've tested them and shown to resolve inflammation. And I'll show you some examples. So this is what we did a few years ago. Myself and a whole host of other colleagues tested some of these drugs. These are the cyclin-dependent kinase inhibitors. And what we showed was that there was a time-dependent and concentration-dependent induction of white blood cell neutrophil apoptosis. So we've shown we've isolated human cells. We've shown that they can undergo apoptosis, but we need to test this in a complex system, like an organism. And what we've done is we've actually used the zebrafish, and I know some of you may have been down to the zebrafish facility. These zebrafish are very small, and we actually work with the embryos, so they're actually microscopic. And what we've done is we've made genetically modified fish with green fluorescent neutrophils. The white blood cells are green and fluorescent, and what you can do is you can visualize these cells under the microscope. We have to induce an inflammatory response, so what we do is we cut the tail fin here, and then we watch what happens to the white blood cells. Essentially, the white blood cells rush to the site here because there's mediators being produced, etc., potential infection in the water here. And incidentally, this will actually regrow again. So regeneration of tissue might be mediated by an inflammatory response. What you can see here is a lot of cells getting recruited, and eventually with time this will resolve. And what Laura and various other people did in the lab is actually made red fluorescent macrophages and cross it with a fish with green fluorescent neutrophils, and can actually look at the interaction of the two different cell types. That's quite exciting. What you can do is do experiments, and Chris Lucas and others in the lab, he's actually looked at this. So this is a control animal at time 0. This is a 4-hour control. You can see lots of neutrophils getting recruited, and after 24 hours some of them move away. And what you can do is you can treat these animals at the peak of inflammation, and you can see resolution occurring. So you can quantify that quite nicely. You can do experiments over time, etc. And you can see that here, if you give the drug at the peak of inflammation, you get resolution. So going back to the lungs, unfortunately, fish are very good at looking at inflammation. However, they don't actually have lungs. So what we need to do is go to a different system. So this is a slide you've already seen. Lung inflammation has lots of white blood cells getting recruited, there's lots of mediators, there's swelling in the lung, etc. So what we did, we went back to a mammalian system. This is a mouse model, where we took a product that's actually liberated or produced by bacteria, we injected it into the lung, and at the peak of inflammation, this is the peak of inflammation, and I'll show you a slide in a minute to show that, you have maximum number of these white blood cells getting recruited. You can give your drug. This is another CDK inhibitor, cyclin-dependent kinase inhibitor, and you can assess inflammation afterwards. And this is what happens. So if you look here, these are maximum white blood cell accumulation. It happens to be neutrophils, and then it resolves. This is a resolving inflammation, a gentle type of inflammation. If you look at the lungs here, you can see lots of neutrophils. These are mouse neutrophils. These are alveolar macrophages. And if you treat the animals at the peak of inflammation with the drug, you can see you get resolution. You have nice resolution occurring. And you can quantify that. If you look at the control, this is the resolution interval, the 19 hours. But with the drug, it's 7.5 hours. So you can actually resolve inflammation quicker by triggering cell death by apoptosis. You can look at the lungs. Here you have your LPS, you have your lovely looking neutrophils. You have some inflammation in the lung. This is the alveolar spaces. When you treat it with the drug, you see less inflammation. And you see cells which are apoptotic. And if you look very carefully, you can see macrophages containing apoptotic cells. So macrophages, another type of white blood cell, can recognize these apoptotic cells and remove them in a silent, non-inflammatory manner. So just a couple of slides left. This is another series of experiments that we do. We take out mononuclear cells, monocytes. They're another type of white blood cells. We put them in culture for about 5 to 7 days. They change into macrophages, these big fried egg-shaped cells here. And then we feed them a meal of apoptotic neutrophils, dead neutrophils, and see what happens. Is understand the fundamental process of inflammation. We think that you can enhance the resolution of inflammation by interfering with some of the biological processes that I've mentioned, especially apoptosis and macrophage removal of apoptotic cells. So this is a normal type response which is resolving over time. Obviously, this is if it's in hours or days or even weeks, but if this is longer in years, it's a severe problem, and actually patients might die and never resolve their inflammation. So what we're trying to do is develop new drugs that you can give to patients at the peak of inflammation when they present in the hospital with an inflammatory disease, especially a lung disease, and actually drive the resolution of inflammation. So we've set out this question here: does selective induction of inflammatory cell apoptosis and clearance of apoptotic cells enhance the resolution of inflammation? And we think the answer is yes, definitely yes. You can do this in a laboratory. They need to be tested in the clinic, and this is where we're going. You can actually do the opposite. You can use pharmacological agents. I'm a pharmacologist. You can use pharmacological agents that do the opposite, actually delay the process of apoptosis, of cell death, and you prevent resolution of inflammation, or you prolong the inflammatory response. So without further ado, I would just like to thank all my colleagues. There's many people involved with this work. Currently the guys in the lab do most of the work at the moment. These are the guys that were in the lab, lots of collaborators, too many to mention individually. I must thank the MRC for funding our research. And because I was talking about apoptosis, and it means leaves falling off a tree, there you have it. Thank you very much.